Welcome to Cell Biology. Today, we are going to explore one of the most fascinating and essential processes inside our cells, the formation and function of coated vesicles. Imagine a bustling factory that needs to package and ship thousands of products every minute without making a single mistake. Your cells do exactly this, using tiny protein-coated bubbles to transport vital molecules. By the end of this video, you will understand not just what these vesicles are, but the beautiful geometry and biophysics that govern how they are built. Let us dive in. Now that we understand the factory analogy, let us look at how the cell actually packages its goods. In the microscopic world of a cell, you cannot simply throw loose proteins into the cytoplasm and hope they reach the right destination. That would lead to absolute chaos. Instead, the cell uses coated vesicles, which act exactly like sturdy, labeled cardboard boxes in a shipping department. These vesicles are fundamental to the cell's internal transport system. They ensure that newly synthesized proteins and other vital molecules are safely packaged, sorted, and delivered to their precise locations within or outside the cell. Building on the idea of shipping containers, let us define what these structures actually look like biologically. A coated vesicle is essentially a small, membrane-bound sac, but what makes it special is the distinct geometric lattice of proteins surrounding its outer cytosolic surface. We call this lattice the coat. This coat is not a permanent fixture. Rather, it is assembled dynamically. Specific protein subunits float freely in the cytoplasm, waiting for a signal. When a vesicle needs to form, these subunits bind to the membrane, linking together to create a protective and structural cage around the lipid bubble. Knowing how the coat assembles leads us to the different types of coats the cell uses. There are three primary types of coated vesicles, each defined by its specific coat protein: clathrin, COP1, and COP2. Think of these as different shipping companies, handling different routes. Clathrin-coated vesicles primarily handle imports, moving materials inward from the plasma membrane, and they also transport cargo from the Golgi apparatus to lysosomes. COP2 vesicles are the forward shippers, transporting newly made proteins from the endoplasmic reticulum to the Golgi. Meanwhile, COP1 vesicles handle reverse transport, catching escaped proteins and returning them safely to the endoplasmic reticulum. With these transport routes established, we must ask why the coat itself is so essential. The protein coat serves two critical functions. It physically forces the flat lipid membrane to curve into a spherical bubble, and it acts as a filter to select the specific cargo molecules that need to be transported. This is absolutely vital for your survival. For example, your brain cells rely on clathrin-coated vesicles to rapidly recycle neurotransmitter machinery, allowing you to think in real time. They also clear LDL cholesterol from your bloodstream. If the genetic instructions for these coats mutate, it can lead to severe diseases like familial hypocholesterolemia, where cholesterol dangerously clogs the arteries. Understanding the biological importance of these vesicles brings us to a fascinating intersection of biology and physics. While cell biology might seem purely descriptive, the formation of a coated vesicle is actually a beautiful problem of geometry and biophysics. The cell must expend precious energy to assemble this protein shell. Therefore, knowing the physical dimensions of the vesicle helps us understand the exact energetic cost of cellular transport. By applying basic spherical geometry, we can calculate exactly how many resources the cell must mobilize to the membrane to successfully pinch off a single transport vesicle. To calculate those energetic costs, we first need to know the size of the vesicle. This formula comes directly from standard spherical geometry and represents the total surface area of the vesicle. In this equation, A is the total surface area, typically measured in square nanometers. The symbol pi is the mathematical constant, and r represents the radius of the vesicle, which is the distance from its center to the outer lipid membrane. Notice that the radius is squared. This means that as the radius increases even slightly, the surface area increases exponentially. A larger vesicle will require a significantly larger number of coat proteins to fully enclose it. Now that we have the total surface area, we can figure out exactly how many individual coat proteins are needed to form the complete protective cage. We do this by dividing the total surface area, A, by the effective area that a single protein complex can cover, represented by the lowercase a. The result, N, is the total number of coat protein complexes required. Think about the physical relationship here. If the area covered by one single protein decreases, the total number of proteins required to cover that same vesicle will increase. This means the cell must synthesize and recruit more proteins to accomplish the exact same job. Let us put these formulas into practice with a concrete example. Imagine a cell is forming a clathrin-coated vesicle with a radius of 25 nanometers. We are given that a single clathrin triskelion covers an effective area of 200 square nanometers. We need to find how many triskelions are required. First, we calculate the total surface area by plugging the radius into our area formula, giving us approximately 7853.75 square nanometers. Next, we divide this total area by the 200 square nanometers covered by a single protein. This yields 39.26. Because a cell cannot synthesize a fraction of a protein, we must round up to ensure complete coverage. The cell needs exactly 40 clathrin triskelions to stabilize this vesicle. We have just seen how surface area dictates the outer protein coat, which brings us to the inside of the vesicle. While the surface area tells us about the packaging material, the internal volume dictates the actual cargo capacity. Vesicles transport three-dimensional molecules, so we must shift our thinking from two-dimensional surface area to three-dimensional space. However, it is crucial to remember that not all internal space is empty and available for cargo. The biological reality is that fluid, structural matrix, and other necessary molecules take up a significant percentage of the interior. We must account for this when calculating how much a vesicle can actually carry. To determine that carrying capacity, we use the standard geometric formula for the volume of a sphere. In this equation, V represents the total internal volume, typically measured in cubic nanometers, and R is the internal radius of the vesicle. Because the radius is cubed in this formula, even a tiny increase in the radius of the vesicle will result in a massive increase in the total internal volume. This volume dictates the absolute maximum carrying capacity of our cellular shipping container. Understanding this capacity is the very first step in determining how efficiently a cell can move materials from one organelle to another. Let us apply this volume formula to a core 2-coated vesicle. We are given an internal radius of 30 nanometers, a single cargo protein volume of 120 cubic nanometers, and we know only 15% of the vesicle is usable space. First, we calculate the total internal volume by cubing the radius and multiplying by 4/3 pi, giving us roughly 113,000 cubic nanometers. Next, we multiply by 0.15 to find the usable volume, which is about 16,964 cubic nanometers. Finally, we divide this usable space by the volume of a single cargo protein.
This gives us 141.36. Since we cannot transport a fraction of a functional protein, we round down. The vesicle can carry exactly 141 intact cargo proteins.
Having calculated the math behind these structures, let us visualize the actual biological process. In this diagram, you are looking at the formation of a clathrin-coated vesicle at the plasma membrane. On the far left, notice the flat lipid bilayer. Specific cargo molecules are binding to Y-shaped receptor proteins embedded in the membrane. Moving to the center, you can see the membrane beginning to curve inward. This deformation is physically driven by the clathrin coat proteins assembling into a geometric lattice on the cytoplasmic side. Finally, on the right, the vesicle has completely pinched off. It is now a perfect sphere, fully enclosed by the protective clathrin coat, with the cargo safely secured inside, ready for transport. We have covered a lot of ground today, so let us review the most important ideas. First, coated vesicles are the indispensable shipping containers of the cell, keeping internal transport organized and efficient. Second, specialized proteins like clathrin, COP1, and COP2 are responsible for physically curving the membrane and selecting the right cargo. Third, we saw that biology is deeply rooted in mathematics. The surface area dictates exactly how many coat proteins are required, while the internal volume strictly limits how much cargo can be carried. By mastering both the biology and the biophysics of coated vesicles, you now understand the fundamental logistics system that keeps every complex cell alive. Great job today.